Hi, everyone. Thanks for joining me in the room today. And those of you that are watching on YouTube later, my plan is to release this on YouTube around the same time that I present it live. And this is a talk that I'm practicing to give to a group of cotton farmers or those who work with cotton farmers that are meeting up in Benin in West Africa. And that's part of the Innovations Club. So I've put their logo on my lead slide here. Um, Really going to talk about nutrient management and different concepts that are discussed around that. A couple of papers that I really like on the subject, but like kind of contextualized around cotton as well, which is actually something that I'm planning to grow for the first time this year, and I'll talk about that a little bit later on. We'll see if I can get them all the way to harvest and learn more about taking care of that plant. Let me talk about the outline today, and I'll mention this picture, which was really fun. It was from Tanzania last year when we went out to a field day in a cotton farming community, and pretty much all the guys that had facial hair wanted to take a picture with me, and a fifth guy tried to jump into this photo who didn't have any facial hair, and we wouldn't let him. Uh, so we were being quite exclusive there. But um, for today's talk, E&M, ecological nutrient management. It's I may default to that. I may always call it by its full name. This is really a conversation around soil biogeochemistry, the interaction of the physical, the chemical, and the biological aspects of soil. And it's actually been quite a while since I was in classes and reading papers all the time about that subject. And so it's been really fun to dive back in. I've been checking a lot of what I'm saying with friends who are actual biochemists and actual soil chemists. And so hopefully I don't get anything too wrong. But of course, with me, before I get too deep into any of the nutrient talk, I want to mention how important it is to have the right mindset around our land management. Nutrient management is part of land management, right? So context is going to be a key aspect of that. I hope that comes through in my talk. It's one of the primary um, aspects of moving towards regenerative land management is to understand the value of context and how things will differ. But always we're going to put the ecology, the relationships of the different organisms at the center of our decision-making process. And it's really going to be about optimizing outcomes. So relative to the amount of resources we're putting in, money we're putting in, time and labor, it's more about profitability than it is about yield, right? And then I will bring up, as I did with my grasslands talk, I just recently practiced the concept of innovation and adoption because it's so central to bringing science to the field, right? And I'll get into some specifics, especially towards the end. I want to talk about a couple of papers that have really got me thinking more in depth about ecological nutrient management, as well as some of a discussion around transitioning our agri ecosystems to this. I mean, that's going to be a theme throughout. So, as an introduction, I wanted to find something that kind of shows the traditional way or the typical way to discuss nutrient transformations. And this nitrogen cycle diagram was what came up to me. I remember learning this in grad school, and I think that there's a real weakness in this diagram in that it just sort of does some hand wavings around the biological aspects of nitrogen transformation and just sort of says like, oh, if pH is here or whatever, then this is the process that you'll get. But that really is cascading through a lot of activities of microorganisms and soil food web organisms in general. So
Yeah.
sorry, I just needed to mute someone there so we could get a clean recording here. So I want to pivot back to, saying again, I'm not a soil chemist. I've tried to check a lot of this with soil chemist friends of mine. But if you ask me too many complicated questions after about the equations and the kind of engineering of these systems from a chemical aspect, then I'm going to probably say, like, let me get back to you on that. Um, However, I will bring up some of the key concepts, including that there's, a lot of the way that we conceptualize of soil chemistry. I'll explain this, but it's by like looking at nutrients in specific compartments and then talking about their transformations. And what's going to happen throughout this is my answer to almost anything will be let's get more soil carbon in there and more diverse soil carbon in there. So, um, yeah, let's go through what soil ecosystem thinking is, embracing like sort of this messy regenerative process. And these are a couple of the papers that I will talk about towards the end. So let's start here. The idea of a nutrient continuum, and I have put an asterisk there because one of my very, um, you know, amazing human friends that I sent this to and asked for feedback, uh, you know, said this is not really the case with all nutrients. You don't have the same continuum, the same process. Is specifically like silica and sulfur were mentioned as being like so there's some caveats in here. This is more common for sort of the nitrogen, phosphorus, potassium, and some of the other key micronutrients we talk about in agriculture. And the big picture here is that the largest pool or a compartment of those elements in soil is typically really quite inactive or unavailable to our plants. It's tightly bound chemically in minerals. It's inside of organic materials where they need to be decomposed or the organisms need to die before the nutrients will be released. And so there is a way for a particular element to switch between that highly unavailable pool into typically like a smaller intermediate pool. It's more loosely bound in the minerals. Maybe it's been a little chemically weathered. There's been some other transformation. Or the organic materials are starting to break down, and instead of a chunk of like undigested wood, which would be unavailable, completely unavailable, they're now in a like partially digested, becoming part of that general organic matter pool, right? That's still not very available to plants in a short term. For that, you really need to get all the way over into this active pool that we talk about being in solution, like in the water within soil, and then on exchange sites as well, right? There are places within the soil where on the mineral surfaces or the organic surfaces, different elements can stick to the surface and then pop right back off when acted upon by, plants give off a lot of acids and things to try to solubilize, we call it. And so that's the big picture here is we've got this unavailable pool that sometimes we talk about immobilized nutrients all the way down to this available pool, and there is transformations happening between these settings. So I want to first talk about soil nitrogen because it's an easy one to point to and say this is important, right? This is important for us, for the plants, for the animals that we have in these systems. A huge component of chlorophyll in plants is nitrogen. So if the plants are not taking nitrogen up from their environment or fixing nitrogen with the help of symbiotic bacteria, then they will not be taking full advantage of the sunlight in the system, right? You have that a lack of photosynthetic efficiency we sometimes talk about. And this goes all the way into like when we talk about anything with DNA, nitrogen is a component of that. This molecule figure on the far right here is actually lysine, which is an essential amino acid. you don't really have proteins that I don't know of any proteins that can be built without nitrogen somewhere in the process, right? It's considered an essential component. So that's what we need to remember if we reach towards these bigger frameworks like One Health, where we're looking at, okay, human health is important, animal health is important. What about environmental? Without below ground life, there's really not going to be above ground life, right? The nitrogen is being grabbed by microbes, by fungi, by protozoa because it's essential to their function, and then that builds all the way up to plants and animals and humans, and then through decomposition, it cascades all the way back down, right? It's linked. But, this is a very complicated thing. It's so hard to talk about soil nutrient management because there's a lot of exceptions. There's things that I would reach for and say, it's kind of like that nutrient continuum. Somebody that really knows what they're talking about will say, that doesn't, that works for 80%, but not everything, right? Well, there's all these processes and attributes of soil, and things get really complicated, complex in there. It's hard for humans to understand. It's very mathematical. I'm not really a quantitatively brained person. Um, my stats classes in grad school were the hardest for me to get past. But I do really love soil. I'm enamored with the story of soil, and that's what I really want to do is make soil nutrient cycling more narrative in our minds, just as long as everybody listening understands that won't be perfect. That you could take something out of context and say, this is my caveat. You could take something out of context and say, well, Adam said X, and that's wrong because over here in this one case, it doesn't work that way. Okay, fair enough. You win. I want to start off by talking a little bit about Hans Jenny and some of the work that was done to identify these 5 key factors that influence soil formation, because they have also a huge influence on nutrient dynamics in soil. And I also want everybody to think about, if you've heard of those 5 factors, which we'll go into, what about the 6th huge influence on soil in the world? So here's our pictures that represent each of these things. I'll bring up the labels in a second. I want people to think about what I'm trying to say here with these pictures. How are each of these things going to be an influence? And again, if you've taken a soils class at a university, you may have already talked about these soil forming factors or soil influences. And humans in the top left are sort of the 6th one that's often left out, but I would say have a huge influence on 80–90% of the soils on this planet. Either a direct influence through cultivation practices and land management, or an indirect influence through, other human activities. Even mining is a great example of something that can influence the soil around it, right? But most of us have thought through about organisms. We know that, for example, worms have huge transformation effect on materials in soil. Time, of course, has this top-down influence that you see soil change. You see the biological community change over time. We call that succession. Relief, or sometimes it's called topography, right? In general, soils end up looking quite different at the top of a hill versus on the middle slope versus at the bottom. And I've actually done some work with researchers in China who were specifically looking at the way the organisms were different across topography and how grazing had a different influence depending on the relief or the topography of the area. Climate is another big top-down factor. I think most of us know that very arid systems have different nutrient dynamics than very wet systems, right? Tropical wet systems often having a really rapid cycle of organisms and nutrients. And as you go towards colder and drier, you often slow that down, right? You see that process very different. And I do note that sometimes in sort of soil enthusiast circles, soil biological enthusiast circles, the influence of parent material is sort of just brushed over. And I think that that's a real shame because the actual chemical transformations that happen between these pools of nutrients can be very different based on the mineral um, contents of soil. If you have soil that was from a volcanic origin versus soil from a sedimentary or a living process, the chemistry can work out very differently, and we might have to manage those soils extraordinarily differently in order to get the outcomes that we need. So it's worth mentioning all of these are important, and this is how I filter chemistry in soil is I think about what are these big influences on it before I get to all of the stoichiometric math of the biochemistry. And I'll just go ahead and say what my bias is. I find the story of what humans and soil life can do together to be one of the most fascinating, right? It's really neat the way that climate and topography and other things can influence soil over time. But once you bring human management of these systems into it, and once you bring sort of that story of whether or not you are setting a stage for less and less soil life or more and more soil life, that's a fascinating place to examine. And I just want to tip my hat here metaphorically to Dr. Elaine because this is probably one of the biggest influences that she had upon me and my thinking during the 3 years that we got to collaborate together. I got to learn from her perspective, and I've mentioned a couple of other individuals who have developed this framework. It's often rejected at agricultural universities to talk this way. It doesn't fit the paradigm, the mindset of how soils classes are often presented. But I think Elaine is onto something to emphasize this. It's drawing that comparison or that contrast between a really like a living soil ecosystem and then dead dirt, degraded dirt. So in living soil, you have this really high quality habitat that facilitates a lot of organisms to be successful in that system, right? To not go into dormancy or death. Whereas in dead dirt, you're kind of treating the ground as a substrate to grow plants. You're focused on the plants, not the microbiome. And as a result, it makes space for these aggressive species, things that we would call pests, whether that's insect pests or microorganism pathogens, right? They're what sort of fills up that environment. And the things that are aggressive and capable of dealing with the disturbance of tillage or something are what persist and succeed versus this really diverse and thriving ecosystem of organisms. As a result of that, Elaine would say, in living soil, not only do you have minerals, but you have lots of air and water. And often when you get into degraded dirt, you can say, oh, here's a compaction layer where we actually don't have the movement of air and water through that. The minerals have become so compressed because the organisms are not there keeping it fluffy, keeping it tilthy, making sure, the development of that soil sponge which is really driven by organic matter as well. It's the food, it's the habitat for those microorganisms. So in living soil, you're going to see a substantial amount of organic matter that affects things like water holding capacity and really helps to facilitate this resilient environment where if a drought year comes periodically, the whole soil environment doesn't die or the plants don't completely fail. right? Or that animals don't run out of food. Um, that's what happens over in dirt, where you have minimal organic matter, minimal capacity to store water and to facilitate the living component of soil. You end up with this very fragile environment. And more human interventions, because humans are a major influence on soils and dirt, more and more human interventions are required to keep that system going, right? Because of its fragility. So, I mean, I would say this is the biggest shift in my mindset coming from an academic setting and then working with Elaine is, I see the world through this lens. Of course, it's a gradient. There's living soils that are slightly and substantially degraded down through the gradient, and there's dirt or things that are dirt adjacent and then improving a little bit and then kind of in the middle and then closer to soil but still dirt-ish, right? So we have to keep that in mind. But again, to do this contrast, I took this picture of these bison. One of the things that blows my mind is to think that at one time there were 40 to 60 million of these relatively unmanaged, influenced by indigenous North Americans, but the systems with very little external input and management, maybe a little fire or something, they developed such a living soil ecosystem. The nutrients were so efficiently cycled that you could facilitate a huge number of animals without tons of fertilizer or irrigation or anything, right? That's the power of living soil. Contrast that to this real picture taken in Texas during the Dust Bowl. Dust is dirt, right? It's the mineral component of soil without the life and the stabilizing influence. And really, the Dust Bowl happened when the people were moving, westward and tearing up that native prairie where there was real soil and getting a few years of amazing crops out of the stored nutrients and other things. And then the destabilization that happened there turned it all to dirt and it blew away. And by the way, this is something that's still happening today. I just saw a news report the other day that central United States has been losing a ton of soil because it's turned into dirt and then dust and then blown away out into the Atlantic Ocean. So, here you go. This is my favorite picture's worth 1,000 words. This group, this Soil and Water Conservation District, they did what's called a SLAC or a SLAKE test, where they took some substrate, they took some material from fields that were treated well, that where tillage was reduced and other things that degrade soil had been sort of corrected over time. And then the neighbor who hadn't done those practices, they took a sample from their land. And when they put them in water, you can just see this is soil, this is dirt or dirt-ish, right? Becoming dirt. It's amazing. It's amazing. It didn't take very long for the water stable nature of soil to come into focus. And part of that is because when we increase soil organic matter every 1%, we can see a substantial increase in water holding capacity. This has been discussed a lot by the FAO and other groups. So, I think this is worth bringing the message home. What's dirt? What's soil? And, I always love to give a plug for Nicole Masters' book. I think especially if you are a producer working with producers, this book should be on your shelf and should be referenced often because it makes the case again that that managing soils looks very different. It's a different mindset that's required. And understanding, having a meaningful sort of relationship with the organisms that are in a living soil ecosystem is very important. If we don't do that, well, I'll get into that because new research indicates that something like 60% of global species live in soil, right? The vast majority of our fungi, even, of course, plants, even things that we don't often think about, little mammals and stuff, they can be found in soil habitat, right? And if the habitat is degraded, and if we lose the organic matter in those systems, it will cause a filtering of the life that can be found there. And I've mentioned before, biodiversity is strength, is resilience in these systems. So, I mean, it's worth checking out this paper. That's why I've cited it here, because you just get a different bird's eye view of what the opportunities are. If humanity sees ourselves as the caretakers and custodian of soil as habitat, we will also get amazing nutrient cycling. If we selfishly mine that habitat for the extractive things that we can take from it, it will degrade. It will be we will have a loss of biodiversity, and ultimately our yields will be less stable, and the whole system will be in that fragile state that I mentioned earlier. So I have to mention here, just biodiversity isn't just for us nerds who are excited about it. Aldo Leopold was a scientist, a poet, and a farmer, and considered by many to be the father of restoration ecology. And I think back in the 30s, 20s and 30s, he really started pondering these things and the effects. And this is one of my favorite sayings of his. The first law of intelligent tinkering is to save all the parts. If we say to a bacteria, fungi, or archaea, or a virus in the soil, like, what good are you? We don't need you. Like, we'll change this system in a way that you get filtered out. I think that's a problem. I think we could find out years from now that we've left important pieces out. He used a metaphor of like a watchmaker, and how if they were fixing a watch, every piece would be preserved because they couldn't imagine, that the watch had parts that weren't necessary. And so now, if we think of the watch metaphor in land management, every component that system evolved with, um, it has a role, it has a function. We know that, or it wouldn't have been preserved through time. Okay, so here we are to holobionts. And one of the reasons why we have a Holobiont hub. Uh, it's not thinking of things just as like, say, a cotton plant in this case, or a tomato plant, as the picture shows, not managing just an animal or a plant, but recognizing that huge microbiome, all the other organisms that interact and share potential pathogens, mutualists or allies. Even there's a ton of just commensalists or neutral players that live in and on the surfaces, and inside of our plants and our animals and ourselves, right? We humans are holobionts as well, a host organism that has a very complex microbiome. So, as some people have talked about the idea of CSA, or climate smart agriculture, they have developed that there's this idea of three pillars. And it's pretty fun that if I were to imagine the concept of holobiont smart agriculture becoming mainstream, it would also have three pillars. You have these active allies, that part of the microbiome we need to have, their presence, their abundance, their diversity of the biological community, not just mutualists, but also things that take up space while doing no harm or no benefit. And not just microorganisms, but earthworms and all of the other creatures that interact with our plant hosts, right? You need them. If they're gone, you're going towards dirt. If they're coming back, you're going back towards a completely thriving soil ecosystem. We also need the host, the plants, animals, to be effective as hosts, to be good partners, right? And that comes into how have we bred plants and animals, especially over the last few hundred years. I got to speak with a dairy operator in Oklahoma where I said, "What's the biggest shift that's happened in a positive way in the last decade in your business?" And the first thing that he said was, "We've learned to breed for cattle health, not just milk production. Because if we have healthier animals, they'll give us more milk across their lifetime. We'll have to do less, you know, euthanasia of animals that go down because their hips aren't strong or some other horrible disease comes in there that's expensive to fight." And I was like, there we go. He's made his dairy cattle, he's optimized them as a host and as a unit of milk production, right? Whatever you want to look at that as, um, by breeding for a more holistic idea than just yield. It's a great example. And of course, we have to maybe when I said that we humans are partners with soil life, the coolest thing we get to do really is make sure we support them, create an environment that, it's not too wet, it's not too dry, if we can get away with that. Or we don't leave a lot of the ground uncovered for a lot of the year, or a lot of the ground productive, right? Fallow, because we know that that doesn't support these thriving microbiomes. So we get to make a lot of those decisions and bring those strategies into employ that can either cause degradation or regeneration in the system. And why does this all matter? If we can find this sweet spot where all three of these things are true at once, then that's where resource use efficiencies, pest and disease suppression, optimized yield and quality, and the resiliency of those systems can really develop and thrive. And if we achieve this, what I put forward is the idea that it can get easier, not harder. There may be an adjustment period where it's pretty tricky to get all of this going at one time. But once it's up and running, once the system has shifted, uh, nutrient management becomes an easier target to hit, right? So, and we'll get into that towards the end. Now, I've thrown a lot of theory and ideas at you today. I want to remind everyone that I'm not just an egghead who went through his time in academia and didn't connect with producers or didn't see the way that things were done at a large scale. I participated in Oklahoma Ag Leadership, Class 19, and spent a year and a half traveling to all these different places in Oklahoma, seeing what conventional agriculture was doing. That's me smiling as I'm looking at a huge field of sorghum in the panhandle of Oklahoma, which is one of my favorite plants. There are many producers, including a cotton farmer, in that class of people. And so having discussions all throughout these seminars and field days and, some of the best stuff happened in the evenings when we were having a beer together, right? So this also informs my perspective, and I commonly look at things and say, okay, that's fun, that's cool, dirt and soil, I get it, but what does that mean practically? So I want to get into some of that. I mentioned that I want to grow some cotton. It's interesting, I'm going to probably have to keep the cotton plants inside, maybe somewhere here in my room for at least a month, because the nights are too cold still in June where I live. And so if I start them indoors in April 15th and they've emerged, 4 to 9 days later, if I put them outside, it could still drop like below the threshold. I think that's about 12 degrees Celsius where they would get harmed, right? And that's just until July and August here, we don't have hot enough evenings for something like cotton, even though we can have hot days. But in general, harvest often between 130, 160 days. My understanding is 130 is the more normal, if everything is kind of going right with the cotton. I should be able to harvest it before things get too cold here in September or something like that. Um, I'm gonna I'm gonna give it a shot. If I'm speaking about with cotton farmers, I feel like I should know more about the plant. And so again, this is sort of my model for today's talk. And I wanted to talk about nutrient management. So the plants will show us if they need us to do something about nutrients. Of course, it may be too late by the time they show us. This is an example of cotton chlorosis. Often, especially if it's pretty uniform across the leaf and across the plant, it reflects that there's a nitrogen deficiency in the plant. It, again, doesn't have enough nitrogen to make enough chlorophyll to maximize its photosynthetic ability, which can affect plant height, eventually yield. One of the interesting things when it is nitrogen deficiency is I understand that later on in the development of the plant, you can actually end up with a reddish tint to the leaves as well. And that could present to you as a different mineral that might be missing. It's actually nitrogen. And of course, I'm not an expert in this, and thankfully, the folks I'm speaking with have access to people who truly are cotton experts, who can say, "Oh yeah, if you see it on the fifth leaf, it means this, but if you see it on the first leaf, it means that." And it's not always nitrogen deficiency, but it often is. And one of the ways that we, sort of in the conventional agriculture world, think about dealing with that is if we see signs either from last year's crop or early in this year's crop of yellowing, chlorosis like that, it means that we need to adjust our strategy around the 4 R's of fertilization, right? That's the source, the rate, the time, and the place. And so there's all this really cool work that has been done to say the first peak demand is before 60 days ish with the crop, before the first bloom. The plant needs to take up about 30% of its total nitrogen. So we have to have the concentration in the soil at this level. And then we got to get in there usually again with a second application. And between 60 and 80 days, it's taking up another 70% of its nitrogen needs, right? This is like when bulbs are starting to develop, or sorry, when blooms are starting to peak on the plant. And so this is pretty costly. This can represent a lot of fuel used to apply nitrogen fertilizers. Nitrogen fertilization is one of the more expensive things that my producer friends tell me that they spend money on. And, it is important for us to not just say, oh, magically nitrogen will work itself out if we have a lot of soil biology, because there are different peak demands in most crops. And there are times we may need to add something in order to bump up the amount of nitrogen that can get into our crop at a key stage. Now with boron, which is another often big problem in cotton development, the signs look really different. Like these little concentric rings might form on the stems, and it's most common in young plants, but they may start aborting flowers, which can affect yield in the long run if they're not taking up enough boron. It's a very critical element for cotton production. And it's something to note that, again, when it comes to mineral nutrition of plants, there are certain things we can add. Phosphorus is a great example, where if we get the rate too high, it can interfere with the plant's capacity to absorb certain other minerals. And I wonder sometimes if the signs of boron deficiency, people would think, therefore we add more boron, but actually it's something else really out of whack that is causing the plant to not be absorbing boron. Now, one of the ways that this is addressed occasionally, at least in high production systems, is to look at, especially this key flowering through bulb development phase, is people will put some foliar applications of boron. And it's a really interesting chemistry because boron tends to stick to cell walls and become embedded in them, is my understanding. And so it's harder for the plant relative to something like nitrogen to move the boron around its own tissues. It tends to stay put. And you can get into some high expense and very low like yield changes when you're talking about foliar applications of mineral fertilizers. It's just something to be aware of. This is how conventional agronomy would say, like, we need to address this deficiency. Or really, are we just talking about burning money again, right? This is the thing. If the system takes these huge inefficient inputs of fertilizers, commercial products every year, and you're tuning that system to my yield needs to be really high to recoup those costs. And is there another way to go about this kind of nutrient management? Well, one thing I want to mention that has been talked about almost in every nutrient talk since, the last 50 years probably, is soil pH. So we know that certain minerals, certain nutrients become relatively more or less available to plants depending on the pH of the soil. And my understanding is that cotton likes this very slightly acidic category. Um, so down to slightly acidic, right? The problem here is soil is not homogeneous. So I don't know of any system, any test that doesn't cost like way too much money where you could really get the optimal pH and level of nutrient added. You can go and play around with things like soil grids. It's free, global. I've used it even in publications before, and you get this idea that even within a few hectares, you can have massive variation in the concentration of nitrogen or something else. And I really don't know. I mean, yes, we need to get our pH managed. And if it gets really high or really low outside of the range of our crop, there are interventions that can successfully work to bring it more into the range it needs to be in. They can cost a lot of money. They can take a lot of time to work. And, thankfully, this is a rep for soil carbon again. Organic matter does a better job, in my opinion, of buffering the effects of soil pH than almost anything. If we can really get that organic matter, if we can add enough and keep it accumulating in the system, it becomes this amazing way to say, oh, over here pH is a little low, but there's enough organic matter to sort of change the dynamics of what's going on with plant chemistry, plant soil interactions. Over here it's a little too high, but again, we have enough organic matter. And many of the research papers that you read about soil chemistry and nutrient management are kind of they're making the assumption that you're working in dirt and not soil. And that's another huge thing that Elaine has given me, is this freedom to say maybe something like 80 or 90% of the evidence that people rely on is actually not reliable unless you're talking about dirt farming. If you're talking about soil farming, it changes that dynamic and things get a lot, getting the pH. I'll also mention, EH is something that's been talked about a lot. The different, it can be buffered by how much organic matter you have. Because healthy soil is going to be crummy. It's going to have lots of these aggregates that are facilitated by the living portion of the soil and the organic residues that are in there, right? And aggregates are really hard. This is soil crumbs, it could also be called aggregates. but it can be really hard to visualize. This paper is probably the coolest I've found to help visualize this. So if you're to pull off what might be called a macro aggregate from a soil system, a larger soil crumb, it could even be 1, 2 millimeters in size, visible to the eye. Sometimes they're called water stable aggregates or macro aggregates. If you could examine this down to the it's a whole universe, right? It's a whole world. There's going to be particulate organic matter stuck in different crevices, right, of this. There's going to be pores. There's going to be fungi and plant roots, and you might be able to actually say, "Oh, here's a grain of sand, silt, or clay that we can separate." But it's all stuck together with biological glues and other factors. And then you get down to the micro aggregate scale, and that's where you may be actually at like 1 or 2 tenths of a millimeter, right? So now we're in the micrometer scale. You could still maybe pull off the mineral grains. You're going to see that like fungal hyphae is starting to collect these particles together. Bacterial glues, again, particulate organic matter, which is like what we think of as decomposing plant materials, for example. That's going to be a factor there. And you can go even deeper into that, and you will see the way that the organomineral coatings and films are happening to hold this together. The bacterial colonies, now we're at what 100th or 200ths of a millimeter in scale. It's really fascinating to think about. Why do I bring this up? If we're talking about something like pH, do we think pH is going to be exactly the same in this part of an aggregate versus that part? Do we think that every aggregate is going to have the same pH as the aggregate next door, right? There's amazing variation that can happen in scales and gradients, and that's actually good because different minerals and different nutrients are more relatively or less relatively available to plants at different pH. Similar, any other, sort of chemical aspect of what we could study. I just think we often have this mentality that if we go take a few soil samples in a field, we can get this snapshot of what's going on. But what's happening is happening down at, a millimeter or two in scale, or even smaller. So I just wanted to put that in there to help us all open this perception in our minds of you know the soil environment and what it's actually like when all of these things are interacting. So you might ask me then, okay, Adam, how do we manage these things? You've brought in all this complexity with aggregates and things. How do we get there? As far as humans and setting the right environmental conditions and stage, it's going to come back to soil carbon. And I highly recommend you look up this paper where I took this figure because it shows how carbon moves into these systems and how plants can produce complex carbon compounds like lignin. They also produce a lot of simple compounds like the sugary exudates from their root systems, which inside of the microbial community and all their interactions can stabilize, right? Normally we think of simple carbon compounds as going into soil and popping back out as CO2 really rapidly. However, microbial necromass, the dead bodies of microbes, and especially fungal um, um, actions and activities and metabolism, the entanglement that the fungal hyphae do of particles can protect that carbon, can make it persist inside of inside of an aggregate, especially a macro aggregate. It can persist for decades, maybe even hundreds of years until something comes along and breaks that aggregate open, gets into the sort of core of that particulate organic matter, and is able to start decomposing it, right? So this whole question about the balance of carbon effluxing out usually is CO2 from microbial respiration and plant respiration and insect respiration versus persistence. This is the way we can really buffer these systems and manage the nutrients in a different way. Um, how do we increase aggregates? Well, when I was in Tanzania, I got to do a field training on pit-produced biochar. And I think biochar has interesting potential to facilitate soil aggregation and microbial habitat. Um, I especially liked it in this context, and I want to give a shout out to doctors Keshav Karanthi and Sandia Karanthi for teaching us about this. In this environment, there is a waste product of cotton stalks. You can see them on the side there, which typically are burnt above ground. And so all that carbon is just released back into the atmosphere. And some of the minerals, maybe 4% of the material, can be, collected as ash and put back in the system. But burning these stalks in this kind of pit, this conical pit, where there's low oxygen down at the point of real combustion at the bottom, instead it preserves a lot of that carbon into the material. And then you take something that would have been thought of as a trash item, and it now becomes a treasure. If you put it out over your field, you can increase water holding capacity and the development of aggregates as if you're getting several years ahead of organic material accumulation. There can be issues with using biochar, including that it can change the pH of the soil system. One of the things I loved that the doctors Karanthi and Karanthi taught us is that you can also gather up weeds from near your cotton field and put them in an anaerobic digester with like an effective microorganism, bokashi sort of condition, and that material will become acidic as it builds up lactic acid bacteria. And then you can combine the two things and get back towards this, maybe 6.8 that is ideal for cotton. So they've really done something here. I would mention there are some detractors of biochar, and I don't consider it a magic bullet by any means. But there's some folks that say, oh, you can actually get environmental pollutants if your biochar production process isn't done just right. However, it was pretty striking to all of us who participated in this field experience that this would have been like a wasted resource, two wasted resources, the weeds and the dried cotton stalks, that now could become actually a meaningful amendment or input. I'll also give a shout out to worms here. They do an amazing job of aggregating materials. It's one of the reasons I use certain worms in my composting system to increase and improve the capacity of those materials to aggregate. And I'll let you read more about earthworms if you're really excited about them. But as we move towards the back end of my presentation here, I just want to rep again for holobionts, but especially fungi. If we can shift that soil ecosystem towards a fungal dominance, then we can start building up more and more stable forms of carbon that stay in the system instead of just going back out as CO2. So bacteria, which do a lot of wonderful things, including liberating nutrients for plants to later absorb, they're relatively inefficient at taking carbon inputs from plant litter or exudates and then locking them into the soil ecosystem. They tend to just breathe a lot of that carbon back out into the atmosphere. However, fungi, if we really have a robust, diverse community as part of our holobionts that we're managing, they can do an incredible job of facilitating the I won't quite say storage, but the retention of that carbon in the system for, again, decades to even centuries. in how they interact with the mineral components of soil and entanglement, and in the kinds of complex structures that they themselves make. They make chitin and other things that are relatively resistant to decomposition, right? So I'm going to bring it back to holobionts here. And I mentioned this soil organic matter value earlier. I'll just rep out again for my favorite fungi, which are arbuscular mycorrhizal fungi. You can read this paper about, you know, how they improve plant nutrition directly by extending the root systems of the plants they associate with. They would associate with cotton in general. And so these limiting resources, including water and other minerals, can be delivered to the plants, improving their photosynthetic capacity, getting more carbon into the system, but also their physical structures help with aggregation. They produce a lot of bioglues, right, that can hold soil particles together. And so if you're having trouble, you think you put a lot of organic matter on your soil and then it decomposes. You put a bunch more, it decomposes, right? You get trapped in this like treadmill of carbon additions. You probably need more fungi. They seem to be one of the mechanisms to shift those systems from carbon losing to carbon accumulating. Okay, so because I mentioned this in the other talk I just gave, I'm not going to spend as much time on it today. I am going to talk about this live with our folks that work with cotton farmers, because I think it's very important to understand any change in land management that we know could work. We also need to filter through human decision making. And so we need to have effective ways of diffusing knowledge. In this case, we've got a few million small scale farmers, many of whom have not completed even a primary education, right? And so we've got to make knowledge accessible to the decision maker. If we, as the change agents who are bringing in new land management techniques, there's a persuasion. Ultimately, that decision happens with the individual. And then I think we really need to be with them every step of the way of implementation, so that it can come back and confirm to them, this actually worked. I got a higher yield of cotton, or this year when there was a drought, all of my neighbors didn't produce anything, but I still did because I had worked on my soil organic matter, right? So very important aspect of the social science to remember here as we talk about all the cool ecology. And this is how this happens within a larger social group. We may first have to spend a lot of time and energy on those 15% of innovators and early adopters and make sure that they succeed so they stick with it. And then eventually, this early majority, late majority, that's 2/3 of the population together, when they can see it, when they can observe that it worked, they're more likely to adopt it themselves, to trial it themselves, and if they succeed, to continue to do it, right? This is how we change the world from a degenerative soil paradigm or mindset to a soil regeneration mindset. And it doesn't happen all at once. Everett Rogers talked about how it often takes 20 years to get this kind of change, but we're not starting from year zero as a movement, which is good news. Okay, I promised I would talk about these two papers, and I will speed up here a little bit just for the purposes of the video, because I'm going to actually have a couple of days to do Q&A with these folks in Benin, and the first day I'm throwing a lot of ideas at them so that we can hash it out over those couple of days. This paper, though, is worth reading. I happen to be friends with the second author, and it's incredible work of bringing together a lot of ideas that are emerging out there in terms of nutrient management and those plant microbe mineral interactions that are happening. So I want to say first that when we talk about organic matter, which I have done several times in this talk, there's that particulate organic matter where there's sort of decomposing, usually plant materials. I guess it could be insects or other animals too. But we're really talking about like carbon and nitrogen compounds that are found, enmeshed together, sometimes entombed, wrapped up in an aggregate or something, and we call that POM, or particulate organic matter. This paper really brings up a lot about this MAOM, or mineral associated. Oh, I spelled it wrong. Mineral associated organic matter. I'll need to fix that later. Um, which they say can actually be a substantial component of the nitrogen cycling. So again, we're talking about compartments here, and that's how they've divided this up in this paper. How plants interact with different types of nitrogen, with different forms of nitrogen, right? This is kind of the big picture. And I'll add to that, I've already mentioned plant exudates or rhizodeposition. This can prime the use of mineral associated organic matter, or the storage of more of it when there's sufficient surplus of nitrogen. There are these mutualisms that come in, like mycorrhizal fungi, which provide different pathways for plants to get mineral nutrition. Rhizophagy would be another one that's talked about a lot. That's really cool. But ultimately, with those microbes and how they're cycling and interacting with these things, I want to remind us that necromass is very important. That when that fungal biomass is alive, there's one value that it's bringing to the system, but maybe when a fungal feeding nematode comes and sucks the juice out of that organism, that the body of it that's left continues to do something really valuable in that soil organic matter kind of biological interactions and chemistry that goes on there. So the second figure that I want to bring up from this paper, and I do want to say that when some of these land management conclusions that I'm making or statements, you won't necessarily find them in the paper. I don't want the authors to think I'm speaking for them, but I'm trying to put this together or synthesize this with other things. In general, what they found here is that a lower nitrogen fertilizer input can often result in the buildup of more of this mineral associated nitrogen, whereas high nitrogen fertilizer inputs can actually deplete the mineral associated organic matter and cause ultimately more leaching, more loss. I would call it less efficiency, right? And so that's what I'm looking at with commercial fertilization as a strategy. It's often very inefficient. There's lots of work that shows that what we're applying ends up in the rivers and streams and atmosphere instead of in the plants as protein. And it really fits more of a maximization or yield based strategy instead of a profit optimizing strategy, while being very ecologically disruptive, right? There's fish kills in rivers that we can track into the fields where corn is grown, and where all those extra nitrogen fertilizers just flush into the water systems. And it's quite devastating. So what I'm putting forward here then is I'm saying, let's look more at the organic inputs. Like when I know that my plants need a boost of nitrogen in my garden, when they're showing me signs of yellowing, I'll add things like blood meal to that system. Or maybe maybe a compost that I know is a little higher in nitrogen based on the materials I put into it. There's fish amino acids and other things, right? There's ways to get the nitrogen needs met through not a synthetic source, but through a biological source. Often requires some investment. And again, if people are thinking, okay, do I have to do this every single year in order to get this yield going? Well, no, there can be a drawdown typically of how much nitrogen it takes to keep that system regulated. As we accumulate the organic matter, mineral associated and particulate, over time. And there's lots of contextual interactions here, but I would say in general, I would expect those organic inputs, and based off what they found in this paper, to create a more dynamic, interactive system where nutrients are cycling more efficiently and more rapidly, going into organisms, going into plants, going back into organisms, right? Going into mineral associated forms, coming back out of mineral associated forms. This is why I find this very exciting. And here we are at the other paper, which I don't have as much to say about, but it is called Ecological Nutrient Management, for smallholder farmers. So it's perfect for the context I'm speaking in. Um, and I love the practicality of saying you can really do something if you invest in three practices, three changes in land management. Increase the species diversity in your field. So in the off season when cotton is not growing, what else could you grow? What could you grow as a companion plant to cotton? What could you bring in, like even the weeds, and do something with so that you can apply it and get different kinds of carbon compound into your field, right? Diversify. And they also say we've got to reduce any bare ground or fallow periods, right? Periods where we don't have plants growing. That fits with regenerative ag principles. Keep a living root in the system, or else you're not feeding your microbes. And in the case of bare ground, they actually might be getting cooked in the sun, right? They also talk about the need to bring in more nitrogen fixation, and they talk about legumes, especially leaving more residues, which is where you have something like perhaps biochar has a place in many cases because those residues normally get burnt off to clear the fields, but maybe they can become something that we retain and build up soil health over time. In the short term, when you make these changes, we often see the system produces a lot of that quick carbon. Maybe bump up the organic matter that the bacteria are working on, and then the carbons they're breathing it back out into the atmosphere, so it can be really tough. Soil organic matter goes up, it goes back down, goes up, it goes back down. Because that rapid microbial growth and that rapid decomposition, it often leaves people feeling stuck. I can't get my organic matter up. What is happening here? I'm putting a bunch of compost into the system. I'm retaining a lot of the plant materials as mulch or something, but it feels stuck. Oh, what they say in this paper is that if you shift the system with these practices, long term, you'll get those more stable forms of carbon. They didn't specifically say a ton about fungi, but I'm bringing together some other things I know and saying the fungi have to be a huge part of this. You get nitrogen and phosphorus accumulating inside the organic matter, right? It's like a bank account that grows in those mineral associated forms, especially, and slower decomposition of the carbon inputs that are going in so that you get this performance feedback. It actually becomes easier to manage the fertility because the baseline fertility is, it more what the plants are looking for, right? Is something at least in that moderate range. So if you're topping it up occasionally, the top up takes less. You're also just those improved water dynamics, the water holding capacity. Cotton is often grown in very arid environments. And so every drop of water, the scarce water that falls on that soil ecosystem, I want it to be grabbed and locked away in the soil until plants and microbes start using it. And then just in general, that greater resilience, we've talked about that capacity of the systems to respond to stress, not by breaking, which would be fragile, but by bouncing back, which is resilient. So that's, the biggest part of why I would say like these investments and this land transition or ecosystem transition, it can be a tough sell. It can be a big lift for small scale farmers, but it is the way that we move into a truly ecologically based land management system. So I put up the summary here. This is the part where I would take questions live from my audience. I'm gonna have to trim a couple slides out here, folks, because it got a little long on me today. But I hope you enjoyed listening, those who are in the room, and I'm gonna stop the recording uh, so that people on YouTube can enjoy just this part of it. And then I've got a little more time to chat.